Welcome everyone. My name is Lejla Gul. I'm a third-year PhD student in Earlham Institute, based in Norwich in United Kingdom. I would like to present now a study about the analysis of an inflammation-related pathway in immune cells, using single-cell RNA-seq data combining with network biology approaches. Based on the literature, the NKG2D pathway is described in natural killer and helper T cells. These cell types are responsible, among others, for cytotoxicity and interferon gamma expression. Interferon gamma is an immune interferon, and it is critical for innate and adaptive immunity. Aberrant expression of the molecule is related to several inflammatory and autoimmune diseases, such as Crohn's disease and ulcerative colitis. These two diseases belong to the main groups of inflammatory bowel disease, which affects the colon. NKG2D signaling has affected by the Crohn's disease based on the literature. However, in ulcerative colitis, there is no strong evidence about the pathway's role in the disease. The aim of our study is to examine the role of the NKG2D pathway in ulcerative colitis in different immune cell types. For the analysis, we use publicly available single-cell RNA-seq data from healthy and UC colon. The transcriptomics have been measured using 10x splicing. After processing it, based on the published method, gene expression was described in 51 different cell types, not only in healthy, but during ulcerative colitis as well. Translating the genes to proteins, we have built up a cell type specific and condition specific, which means health or disease state, signaling network using protein-protein interactions from Omnibus database. From these networks, we have selected 9 immune cells where we thought that the pathway can have a significant effect. These cells were natural killer cell, regulatory T cell, macrophage, 3 subpopulations of CD8 plus T cells, innate lymphoid cell, cycling and inflammatory monocytes. Finally, the NKG2D signaling network has been created for each cell type based on the expressed genes. During this poster presentation, I would like to highlight only the natural killer cell because this cell type is the main source of the signaling, as I mentioned previously. The same network biological representation can be seen, like by the introduction. However, now the colors refer to the gene expression in different conditions in a dedicated cell type. Red nodes are dominantly expressed in disease state, while green proteins appear mainly in healthy colon. We can find yellow points, which are very special. Those genes or proteins are expressed only in health condition. And black color shows the protein which is not expressed at all in natural killer cell based on single cell data. Currently, this protein is the MAP3-Key1. This network image has 2 main messages. On the one hand, the NKG2D signaling is different in healthy and disease state. There are parts of the signaling which can happen only in healthy case, highlighted with yellow edges on the figure. The other information is that in the NKG2D pathway, parallel protein pairs are appearing. Parallel proteins are translated from homolog genes that have evolved by duplication and having the similar but not identical functions. So these pairs have mostly different expression in healthy and disease condition. Therefore, we can assume that these key signaling proteins are leading to signal through different members during ulcerative colitis. Analyzing the network from a bit closer, we have illustrated the pathway in healthy and UC conditions separately. As it can be seen, in healthy condition, due to the more expressed protein, the signal can flow on huge amount of paths, while in ulcerative colitis this number is a bit reduced. Summarizing the results, there are pathway members which are not expressed in the cell, like MAP3-K1, and there are proteins which translated only in healthy condition. These are the condition-specific proteins. These two diverse things can cause a variety of paths between the receptor and transcription factors. It is important to mention that limitation of the results is that the NKG2D receptor itself cannot be found in the single-cell dataset, probably due to the limitation of the 10x pipeline. In conclusion, we have built up a pipeline for not only cell but also condition-specific pathway analysis. We showed that the signal is spreading differently in ulcerative colitis compared to healthy condition. Besides, in the study we found that the expression of pathway members differs among the 9 immune cell types. We have identified parallel proteins in the network, which are differently expressed among the cell types, and the paths show contrary expression between conditions. I would like to thank all of the people who were collaborating in this project, and for the Knowledge Research Park and BBSRC for supporting my PhD. And thank you for your attention.